Hello everybody, we're going to go over test 3 from fall 2013. I already went over the first third of this test, first 14 questions, on the video called Glycogen Extra Help Unit 7. So you'll find the answers and slide references to everything for glycogen there. We're going to start with gluconeogenesis, which is Unit 8. Number 15 is just a conceptual question about what stimulates gluconeogenesis, and the answer is low blood glucose. That's the whole purpose. The liver's making glucose for the rest of the body. High insulin is going to stimulate glycolysis and inhibit gluconeogenesis, and glucagon and epinephrine are going to both stimulate gluconeogenesis, but if they're low, glycolysis will happen. So if either of these said high blood glucagon or high blood epinephrine, those could be answers as well. And we haven't talked about nitric oxide at all as far as regulation goes. So you know the drill by now. Unit 8, slide 32 will be the format of everything on the left-hand column here. The conversion of this metabolite to glucose does not require biotin. So on slide 32, you're going to see the families. Aspartate and asparagine are both in the oxaloacetate family, so it skips pyruvate carboxylase. All of these amino acids are in the pyruvate family, so they need biotin, and lactate goes through pyruvate as well. So pyruvate carboxylase is required there. One of these reactions in the liver is stimulated by citrate. All the reactions you can find on 11 and 12, and the regulation that you should memorize is on 38 and 39. Know that regulation really well. So the one stimulated by citrate is C. FBase1 is the enzyme for it. It's doing the reverse of glycolysis reaction number 3. So make sure you know the regulation really well on that. Glucose precursor is generated by red blood cells. Hopefully you could have answered that one. It's from Unit 3 when it talks about all red blood cells go to lactate, and the lactate's converted and turned back into glucose. Brings it up again in this slide about the Cori cycle. This is really the only thing you need to know about it, as far as I've seen on previous tests. Which enzyme is pyridoxine dependent? So it's B6. This is the only time we're going to see B6, and it's with the 2 aminotransferases. Either aminotransferase would work in this case. Dehydrogenase is niacin, PDH, 1, 2, 3, and 5. All the B vitamins, thiamine, riboflavin, niacin. The other one I'm drawing a blank on right now. Another dehydrogenase and aldolase, no vitamins. So it takes 12 enzymes, 2 vitamins, and 6 ATP equivalents to convert this molecule to glucose. He's going to ask things like this. This is coming off slide 36, where this really shows if you have a full understanding of gluconeogenesis. So the answer to this one is lactate. Remember, there's 2 enzymes, lactate dehydrogenase and pyruvate carboxylase, and then the 10 enzymes of glycolysis, essentially, going up through glycolysis. So let me rephrase that. It's going backwards through glycolysis. Most of the enzymes are the same, but you're bypassing the regulatory steps. So revisit this slide, and there's all of these are all mapped out. And make sure you could trace each one, talking about the enzymes, what vitamins are required, and how much ATP is required as well. For instance, glycerol is the lowest amount of ATP required. One of these enzymes is gene-induced by glucagon. It's There's a straight memorization off the regulation slide. PEP carboxykinase. These molecules can be converted to glucose without a carboxylation reaction and without an amino transfer reaction. So slide 36 again. This is talking about the pyruvate carboxylase. This would be either of the amino transferases. So this one would require the carboxylation reaction, and those two are going, or, all of these are going to require a amino transferase because they're amino acids. These would require both, because they're in the pyruvate family, both a carboxylation and an amino transfer. This enzyme is a ligase. Remember, ligases use ATP. It's pyruvate carboxylase. Other thing to know, that's your biotin one as well. So it's using ATP and biotin. Ligases are enzyme class 6, you could ask it that way. Transferases are class 2. And any dehydrogenase is going to be a class 1. Class 1, also kinase independent. Which of these molecules cannot be converted to glucose? Leucine and lysine. These are two ketogenic only amino acids. Everything else can go through gluconeogenesis. Slide 32 shows the other 18 amino acids, and these are the two that aren't listed on slide 32, the families. This metabolite is not synthesized in a fed liver. So remember, fed liver glycolysis is happening, so you can recognize a lot of this from glycolysis. This is the feeder pathway from Unit 4. This is the potent activator of glycolysis. So glucose, if your liver's fed, it doesn't need to make glucose. There's enough glucose happening, going throughout the body from what you just ate. This molecule is known to inhibit gluconeogenesis. Another regulation question here. This should pop out as being that potent activator of glycolysis, step 3 of glycolysis. So all of these other ones are activators of gluconeogenesis, I believe. Review on these, the regulation slides. I'm not sure totally about UTP, if that has anything to do with it. But this is, on both of those slides. Which molecule is always needed for gluconeogenesis? So these three are needed sometimes. ATP is always needed. So I guess on slide 36 you're also going to see that no matter what, to make glucose you're going to need to spend a little ATP. So whether it be pyruvate carboxylase using ATP or glycerol kinase using ATP, it's always required. This is for the pyruvate, if it's going through pyruvate only, and this is if it's only an amino acid. Enzyme is niacin dependent. Anything with a DH, so lactate dehydrogenase is going to be the answer there. Alright, on to Unit 9, PPP. Which pathway requires NADPH? This is fatty acid biosynthesis. You see it in one version at the start and towards the end of the unit for what pathways and what organs require NADPH, or you have an active PPP. Which molecule can be metabolized by TK and TA? 918 has all of the reactions. It's not the only place you can find them. There's the individual reactions, but that's just the easiest way to make sure you know what all the reactions are, because there's a couple questions like this. So you really need to be able to write out the pathways from scratch. And once you did that, you would know that fructose 6-phosphate is the answer. Remember, these are reversible reactions. Anything in the non-oxidative phase of the PPP is going to be reversible. This enzyme does not appear to be vitamin dependent. It could be trans-aldolase. TK on this slide here shows it's a TPP enzyme, which means it's thiamine dependent, B1. And PDH and alpha-ketoglutarate dehydrogenase, those are both vitamins 1, 2, 3, and 5. This molecule cannot be metabolized by trans-ketolase. So if you look at all the reactions again, these can be. His answer key says this one, but I think ribose 5-phosphate can be metabolized by trans-ketolase. I think what he meant to say was ribulose 5-phosphate. So I think there was a typo in that one. So ribulose is the only one that's involved in the epimerase and isomerase enzyme or reactions. Trans-ketolase definitely has the ribose 5-phosphate as well. So I don't think there's any right answer. This would be what he meant to say. So there's a lot of clinical stuff at the end of Unit 9. Make sure you know it really well. So this is straight off slide 35. Trans-ketolase is involved in this syndrome. For Fauvism, the answer is C. I believe there's 2 other ones on that slide. This is just one of the molecules that can potentially generate it. So know the other 2. I don't have it in front of me right now. I think it's in the very first line of slide 31. Which enzyme does not appear to have prosthetic groups? So if you're looking at his key, he had marked trans-ketolase, and I think that was an error. I believe the answer is G6P dehydrogenase. Trans-ketolase is TPP, so B1, that's prosthetic group. Pyruvate carboxylase is biotin. Biotin is B7, that's another prosthetic group. Alanine aminotransferase has B6, which is a prosthetic group. PDH complex has B1, 2, 3, and 5. These 2 are both co-substrates, and these are both prosthetic groups. So I believe that's what the answer is, even though his key is saying C. I think there was a mistake. I went back to Unit 1 to double check on that. If you go back to the very first unit on slide 55, it lists which ones are co-substrates and which ones are prosthetic groups. So the regulation is pretty simple. This is the regulatory enzyme of the PPP. There's only one slide on regulation in the whole thing. This is just straight memorizing what's the glutathione reductase reaction. The answer to that is B. So back to the regulation of the PPP. What stimulates it? NADP. The purpose is to make NADPH. So if you have this, they want to convert it into NADPH. Process is catalyzed by heme enzymes, talking about cytochrome P450. It's the heme enzyme that you should really know in this one, and that has to do with the metabolism of ibuprofen, the xenobiotics. I don't know if this is the last slide or second to last slide, but that's where this question is coming from. And last one, another clinical question on slide 34. Heinz bodies are, it's talking about red blood cells, it's dealing with hemoglobin. So I just realized there were no questions on really the modes, but do make sure to review those, maybe on some old tests for how he asks about the different 4 modes and what's happening in each of them. but the clinical and knowing just being able to write out the pathways and that little bit of regulation seems to be the vast majority of Unit 9. So good luck, and I will see you guys next time.